Hello everyone, and thank you for joining me in this new episode of Unravelling T Cell Recognition, Insights from Immunology and AI. My name is Hashem Kouhi and I am delighted to be hosting this series. For today, I am very pleased to be joined by Dr. Nicholas Sgourakis from University of Pennsylvania. Nick really doesn't need any introduction for those of us working at the interface of T-cell immunology and data science, but a brief career overview for those who are new. Nick earned his PhD in biology with Angel Garcia in 2009, focusing on blending simulation and experimental approaches to study intrinsically disordered proteins. Under the mentorship from the Nobel laureate David Baker, he focused on modeling the structures of protein complexes using sparse data and completed his training with Adrian Bax and David Margiolis at the NIH, where he explored uses of biomolecular NMR spectroscopy for understanding viral immune evasion mechanisms. These experiences laid the foundation for his independent research program, focusing on deciphering and targeting the MHC class I antigen processing and presentation pathway. During his 5-year tenure at the University of Pennsylvania and Children's Hospital of Philadelphia, Nick has steered his work toward immunotherapy applications against pediatric solid tumors, ensuring that his discoveries had real-world therapeutic impact. Nick's lab is part of 2 international teams that were selected by the International Cancer Grand Challenge Consortium, aiming to develop T-cell-based therapeutics. His team blends structural biology with computational and protein engineering approaches, aiming to restore immune surveillance of intracellular driver oncoproteins in solid tumors. Thank you so much, Nick, for joining me, and the stage is yours.
Thank you very much, Hashem, for this kind introduction. It's a pleasure to present today. And just very briefly, by way of introduction, as Hashem alluded, my lab really focuses on 2 aspects of engineering novel immunotherapies. One hand involves what's happening inside the cell, particularly the process of peptide antigen repertoire selection. This is a process that involves molecular chaperones, tapasin and TAPPR, that we've spent a fair bit of time of characterizing at a mechanistic and biochemical level. And of course, the second part involves, once we have identified robust peptide antigens and neoantigens for cancer immunotherapy, what is the best approach for streamlining the development of therapeutic approaches using engineered receptors? So I'll be very pleased to share with you some work along those lines as well today. So just to give you an overview of my talk, to this audience, there's very little need to provide an extensive description of the antigen processing and presentation machinery. I'll talk a little bit about existing barriers to translation, particularly with regards to cold tumors. This is a class of tumors that presents very low antigen, making the identification of targetable biomarkers really challenging. And I'll go on to understand how to describe how we can essentially fine-tune the peptide exchange function of molecular chaperones, both TAPPR and tapasin, to restore antigen expression and HLA presentation in these cold tumors, allowing us to identify targetable cancer neoantigens. And I'll describe how we can target those antigens across HLA, polymorphic HLA allotypes in different patients, with the ultimate goal of expanding existing immunotherapies. So, the MHC antigen processing and presentation is a fundamental immune surveillance mechanism which allows presentation of essentially peptide barcodes from any intracellular protein to be surveilled by cytotoxic CD8 T cells. Very briefly, this process involves turnover of all cytosolic proteins through the proteasome and other mechanisms, resulting in a generation of pool of peptides, some of these peptides are going to be transported into the ER lumen through the function of the TAP transporter, where they're going to be met with the nascent MHC heavy and light chain complex, and through the function of this multi-subunit peptide loading complex involving tapasin, ERP57, calreticulin, and other co-chaperones, we're going to get selective loading of certain peptides on the nascent MHC groove such that peptide repertoire optimization can proceed. Peptide-loaded molecules undergo quality control through a glycosylation cycle, and some of these molecules are going to be selected and trafficked further through the secretory pathway, where they're being presented on the cell surface for immune surveillance by CD8 T cells. Now, there's enormous combinatorial complexity in the system, stemming from the fact that there's millions of possible peptide fragments that are continuously sampled by this peptide loading complex. There's enormous biological polymorphism in the HLA alleles in humans. This is the most polymorphic locus of the human genome. And also, there is a massive repertoire of T cell receptors with million specificities waiting to recognize peptides on the other side. So really understanding all these interactions, how selection of particular peptides proceeds, what receptors are going to be recognizing which peptides, is the new frontier of immunology with tremendous applications in immunotherapy. So I'll start by talking a little bit more about this peptide repertoire optimization. And here we have to understand that this process of peptide loading is not driven by ADP hydrolysis one way or another. It's simply driven by the peptide concentration gradient. And we have a strong peptide concentration gradient generated via the function of the TAP transporter, where we have a high abundance of MHC ligands in the ER lumen. But in other conditions, we can have unloading of MHC molecules from their peptides. And this process of loading and unloading proceeds hand in hand, is assisted by molecular chaperones, tapasin, and TAP-EPR, and involves many structural intermediates. So under conditions of high peptide concentration, the process favors the loaded form of the MHC. Under conditions of low peptide concentration, such as, for example, in non-ER compartments, or even in the cell surface, you may have unloading. And this process of sampling, if you want, of proofreading of different peptides is crucial for the selection of immunodominant peptide antigens that will drive a strong T cell response. So we began by understanding this process through using a molecular chaperone, TAP-EPR, to characterize the selection of particular peptides over others. And particular questions that are open is exactly what is the mechanism of loading class 1 molecules with peptides? Does peptide loading proceed via the N-terminus or C-terminal capture of the peptide? And how do these chaperones proofread immunogenic peptides against a massive background of unrelated peptide decoys that somehow also must be sampled within the nascent MHC1 peptide binding groove to select particular immunodominant peptides? These are fundamental questions with tremendous implications for vaccine design and immunotherapeutic target selection that we seek to address using a minimalistic system. So what we did here as a first pass is characterize the interactions of a molecular chaperone, and here TAP-EPR, which is a homologue of the primary MHC chaperone, which is tapasin. TAP-EPR offers a really attractive system for being able to do recombinant protein production, which enables detailed structural and biochemical studies. So here we turn first to using solution NMR as a means of directly visualizing the interactions between TAP-EPR and nascent or peptide-loaded MHC molecules. And appreciate that TAP-EPR binds on the underside of the MHC1 peptide binding groove and also forms interactions with the alpha-3 and beta-2M light chain, but also induces long-range allosteric effects on the MHC1 peptide binding groove. These studies were enabled by our ability to introduce site-specific isotopic labeling of MHC molecules, where we can particularly look at methyl groups to characterize this high molecular weight protein complex. We also characterize interactions with the non-classical MHC molecule MR1, which presents metabolites to the immune system, and found that this interaction also observes the canonical MHC1 or MR1 antigen recognition mode and occurs in a more or less antigen-independent manner. So TAPPR and tapasin utilize a conserved docking mode on MHC and MR1 molecules to enable the selection and optimization of particular peptide or metabolite antigens by maintaining a nascent MHC molecule in an empty ligand receptive conformation. So these studies offered us a glimpse on how chaperones interact and proofread, if you want, MHC molecules. But in the case of MHC complexes, all of these structures involved the empty state of the complex. And all the crystal and cryo-EM structures that were available at the time completely lacked any density for the peptide ligand, which made it very difficult to understand how specific peptides interact with the chaperone complex and how these interactions trigger the selection of some peptides over others. To capture the peptide bound form of the complex, we had to first optimize the system. And here, through a collaboration with Eric Procco and Christine Devlin, at the time at the University of Illinois, we performed deep mutational scanning of the TAPPR interaction surfaces with MHC molecule, which was identified using our solution NMR experiments. And to make a very long story short, we identified a triple variant of TAPPR, whereby introducing 3 amino acid substitutions at the interface with the MHC, we could drastically enhance the affinity, which allowed us to capture interactions with peptide-loaded MHC molecule, as opposed to all the existing structural data which existed at the time involving the empty form. This also translated in a drastic increase in peptide exchange capability. So here you see how introducing these 3 mutations in the high-fidelity TAPPR variant drastically promotes the unloading of fluorescently loaded peptides on MHC molecules in the presence of large excess of a competing peptide. And we see here a drastic enhancement in the catalytic activity of this high-fidelity TAPPR variant. Now, ultimately, this allowed us to capture and characterize the proofreading complex between TAPPR, MHC, and a peptide decoy. So here we see a very short peptide that would never stand a chance of becoming part of the repertoire, but is still captured and proofread by these backbone-centric interactions in the hydrophobic pocket formed by the A and B pockets in the MHC. The N-terminal part of the alpha-1 helix is completely missing. This part only folds when a high-affinity peptide reaches all the way into the F pocket, and this is the trigger. The folding of the alpha-1 helix is the trigger that promotes chaperone release for the complex, such that these longer peptides, which have the right sequence features, can become part of the peptide repertoire. So through protein engineering, we were able to capture these proofreading complex and understand the mechanism of repertoire selection. But really, the key question here is how can we use the system to do something helpful? And here we have to recognize that in the canonical scenario of well-processed and loaded MHC molecules, we have a wealth of peptide antigens presented in tumors. There's good T cell infiltrations. There's good productive immune response and against the tumor. However, in other scenaria, particularly when the levels of HLA expression are reduced beyond a certain critical threshold, we get an exclusion of immune cells from the tumor microenvironment. And eventually we come to a scenario where we have really low HLA expression, where we go to these immune desert tumors with effectively no T cell infiltration and no immune responses. Now, how this actually happens, it turns out that chaperones are again at the center of this. So if we analyze different HLA expression levels, here we see a panel of neuroblastoma cell lines. We see that the number of peptide antigens that can be isolated and characterized from these cell lines strongly correlates with the expression level in these in the same cell lines. So for hot tumors, like up here on the right, we have a wealth of peptide antigens that can be presented and characterized. As we drop below a critical point of HLA expression, there's only a handful of antigens, which makes characterization of targetable biomarkers very, very difficult. So if we look across all these different tumors, and here's just an analysis of data available in DepMap relative to their expression levels in healthy tissues from GTEx, we see that, of course, across a lot of these tumors, we see a downregulation of HLA A, B, and C, which makes a lot of sense in light of what we just presented. However, across all these tumors, there's a very striking downregulation of both MHC1 chaperones, tapasin, and TAPPR alike, which provides a hallmark, really, for low antigen presentation across all these different tumors, which leads us to really the idea that perhaps we can leverage engineered MHC1 chaperones to restore or enhance MHC antigen production in these immunologically cold tumors and to prevent tumor relapse in different immunotherapeutic settings, both for the perspective of discovering new targetable antigens and neoantigens, and from the perspective of invigorating immune responses in these cold tumors. So to understand this, we have to also understand the powers that shape the abundance of chaperones in the ER. So here, focusing on the primary chaperone, which is tapasin, there's beautiful work, really, that has characterized the balance between tapasin targeting to the peptide loading complex, where peptide loading occurs, but also tapasin targeting to this RNF185 membrane ERAD complex for turnover. This is great work from the Carvalho lab here at Oxford. And this appears to be mediated by a lysine, which is found on the transmembrane domain of tapasin, right at the boundary between the transmembrane domain and the cytosolic part of tapasin. But also there's a second double lysine motif that's also implicated both for ERAD and turnover of tapasin, as well as retrotranslocation of tapasin from the Golgi back into the ER lumen. And at any given time, about 5% of tapasin molecules are in fact located in the Golgi. This is the work of Katze-Poulsen back in this beautiful JBC paper here. So the system both has the HLA antigen production has a rheostat, and that rheostat is in fact tapasin. And under conditions where we have tapasin targeting to the peptide loading complex, we're going to have high productive MHC1 expression. Under conditions where tapasin is recycled through this ERAD complex, we have low MHC1 production. So we reason that both of these arms of the pathway can be leveraged for therapeutic targeting with the aim of enhancing antigen presentation in these cold tumors. And we really focused on two approaches. One approach focuses on delocalizing tapasin to promote the loading arm of this pathway. And we do that essentially by replacing the membrane and cytosolic domain of tapasin with modules that allow tapasin to be expressed throughout the secretory pathway to create a continuum of chaperoning activity, guiding MHC1 molecules to the cell surface. And the other approach involves the same deep mutational scanning studies that I alluded to previously for TAPPR, extended to tapasin to identify amino acid substitutions that confer a higher affinity of interactions between tapasin and nascent MHC molecules, again, for the ultimate goal of enhancing loading and peptide presentation by MHC1. I'd like to talk a little bit about the first approach. And we started looking at this by simply taking hex cells and knocking out tapasin, and then on this background, introducing different variants of tapasin, just with lentiviral transduction. And we see here that wild-type tapasin can, of course, restore HLA production in these cells, which is not possible using TN6, which is a variant of tapasin with mutations at the interface with the MHC surface. there's no interaction with the MHC, there's no effect in HLA production. Now, surprisingly, mutations of both the Golgi retrieval signal of tapasin or the ERAD targeting signal of tapasin had very little effect on HLA expression. However, what we found was that when we combined both types of substitutions, now we have a variant of tapasin that has a significant effect in upregulating HLA production relative to the wild-type chaperone. And at that point, we reasoned that tapasin functions just as a classical molecule from the secretory pathway, and we completely replaced the transmembrane domain cytosolic motif of tapasin with that from a generic secretory molecule such as HLA-G. We can use CD8. It doesn't really matter. We'll get a very similar effect. Then when we took this variant of tapasin, tapasin-TM, and introduced it to EBC1 cells, which is a classical coldest of the cold, as we call tumor, this is a neuroblastoma cell and that has very low antigen production, we see that introduction of this tapasin-TM construct massively enhances tapasin expression levels relative to both parental cells or transduction of those cells with a wild-type variant of tapasin. So delocalizing tapasin and armoring it against degradation by ERAD leads to a enhanced production of tapasin and hopefully tapasin function. The next thing that we asked is whether this increased tapasin production translates to increased HLA production on the cell surface, and this was certainly the case. We see here that we get more than double enhancement in HLA surface levels relative to parental cells upon transduction with our tapasin-TM variant, which is not matched at all using the tapasin wild type, and the tapasin-TN6 control, of course, has a similar phenotype. Then we ask, well, how does this ability to restore HLA production depend on the steady-state HLA expression in the original parental cell line? So here we used a range of different neuroblastoma cell lines that have a range of parental steady-state surface levels. And what we find is that cell lines that are very cold to begin with, such as these EBC1 or NLF cells, greatly benefit from introduction of our tapasin-TM construct, whereas cell lines that have pretty good HLA production to begin with have no benefit with respect to HLA cell surface expression. But hold that thought, because we did find that they do have a benefit with respect to the stability of those molecules on the cell surface. Then we asked, well, we know that tapasin has an HLA dependence, and different HLA allotypes have different dependencies on tapasin for loading and cell surface expression. And what we found using our system is that the same preferences are respected using the tapasin-TM system. So here we see that tapasin-dependent alleles like A01 and C07 have a great benefit from the introduction of tapasin-TM on a tapasin knockout background, whereas relatively tapasin-independent alleles like 6801 don't seem to have much an effect as expected based on literature values. So we wanted to delve deeper into the mechanism via which introduction of this tapasin-TM variant allows for efficient class 1 production on the cell surface. So we perform a series of trafficking assays. Here we treat cells with different inhibitors of the endogenous processing and presentation machinery and monitor the rate of decay of HLA molecules on the cell surface upon incubation at different time points. Cyclohexamide here inhibits HLA synthesis. Brefelding A inhibits the enterograde transfer through the secretory pathway. Epoxymycin here inhibits the proteasome. And we see that in all of these three inhibitors, we see an enhanced stability of cell surface HLA molecules upon transduction with tapasin-TM. And this was all done in the cold EBC1 neuroblastoma. We see reduced sensitivity to bufilomycin A1, which inhibits the lysosome system, which suggests reduced targeting of HLA molecules to the lysosome upon transduction with tapasin-TM. So all these effects suggest that the presence of tapasin-TM stabilizes MHC molecules at various steps throughout their processing pathway. And when we apply this to different cell lines, we see that, as expected, cell lines that are cold and benefit significantly from the introduction of tapasin-TM also saw a drastic enhancement in stability of cell surface HLA molecules. But surprisingly, cell lines that are already expressing significant amounts of HLA on the cell surface at steady state and don't show an upregulation of HLA per se upon transduction with tapasin-TM still show an enhancement in the stability of those molecules in the presence of our engineered tapasin-TM variant. So this led us to hypothesize that there's two possible mechanisms via which introduction of tapasin-TM may restore antigen production. One is by conferring an optimized peptide repertoire. After all, this is the function of tapasin. It's an optimizer of the repertoire towards high affinity peptides. Or another possibility is by sending tapasin directly to the cell surface, we could have a direct stabilization of surface molecules through physical association with tapasin on the cell surface. So to answer the first question, we turn to immunopeptidomics. And here we see that, again, using EBC1 cold neuroblastoma cells as a system, upon transduction with tapasin-TM, we can recover a much higher proportion of high affinity ligands on HLA. What I'm not showing you here is at the same time we reduce the background of low affinity peptides or peptides that are not predicted to bind at all to the HLA allotypes present in the cell line. So effectively, introduction of tapasin-TM optimizes the peptide repertoire towards higher affinity ligands, effectively cleaning up our immunopeptidomic data. But at the same time, we see that whereas we can capture a significant proportion of the parental immunopeptidome, and for those peptides, we also can characterize the parental immunopeptidome with much higher counts and much better reproducibility upon independent immunopeptidomics experiments. Introduction of tapasin-TM also helps to reveal about 42% of additional peptides that could not be identified in the parental cells, presumably due to their low copy numbers. So when we go and look at this effect across different cell lines and across different HLA allotypes by assigning peptides from our immunopeptidomics to specific HLA allotypes present in those cell lines, we see that introduction of tapasin-TM benefits most HLAs, really, particularly C alleles like CO701 and CO702, show a drastic enhancement in the number of capturable ligands. And this effect persists upon all cell lines that have low or moderate HLA expression levels to begin with. So we asked the question whether these peptide ligands can provide us a source of target antigens for therapeutic applications. And at the same time, we asked whether the second possible mechanism, a direct interaction of tapasin with HLA on the cell surface, could indeed happen as a means of stabilizing these molecules further. So for this, we turn to super resolution microscopy. These are DNA paint experiments where we can track independently HLA and tapasin molecules directly as they're expressed on the cell surface. So this is a neuroblastoma cell, and these clusters that you see here correspond to individual HLA molecules directly on the cell surface. This is the expression pattern of tapasin. And if we zoom in, we see these clusters where indeed we form hubs where HLA molecules co-localize with tapasin into microdomains. Now, we also performed super resolution tracking of individual HLA molecules in live cells. And we find that the same regions where we see high pair correlation between tapasin and HLA correspond to clusters of slowly diffusion HLA molecules. And we know from the work of Michael Edidin and others how important these MHC1 microdomains and organization of MHC molecules in microdomains is for T cell activation. So this provides very strong evidence that tapasin, when sent on the cell surface, can indeed co-localize and physically associate with MHC molecules to prolong their stability, both for immunopeptidomic capture, but also for T cell activation. So turning back to our immunopeptidomics data, we asked the question, well, can we identify some antigens with suitable therapeutic windows amongst our data? So we built a multi-omics pipeline, considering data both from primary tumors as well as neuroblastoma cell lines, as well as using all our immunopeptidomics data, first on just a single allele, AO201, as inputs, to really develop a pipeline for cross-referencing peptides with respect to the expression of the genes of origin in normal tissue. And we identified several peptides from these genes here that are very low expression in normal tissues, a high consistent expression across multiple neuroblastoma primary tumors and cell line samples, providing us with the right therapeutic windows for targeting. We also ask whether these peptides could be represented in the self-peptidome. So here we leverage a database with peptidomics data from multiple healthy tissues, and we see that these peptides are mostly absent, with the exception of this one peptide here from DBF4. These peptides are mostly absent from healthy tumors, suggesting, again, reasonable opportunity for therapeutic targeting. The peptides here are mostly restricted to AO201 and its micropolymorphisms. However, we did identify peptides that have the potential of being presented across multiple common HLAs, suggesting bona fide public antigens, again, for targeting. So we then made recombinant complexes, HLA complexes, for our top candidates from these peptides. And here we see melting curves, suggesting that, indeed, for a lot of these peptides, we can isolate a stable, high-affinity complex with the MHC. We then panned for T cells, and here we're seeing ELISpods for a couple of these peptides, suggesting, again, that we can elicit T cell responses for further therapeutic development. So all this data are just based on one allele, AO201. We then expanded across 6 different cell lines, across all alleles present in those cell lines. And here you see, again, how many more additional peptides we can reveal and analyze from those cell lines. We combine the data together from the parental and the tapasin-TM transduced cells, and when it's all said and done, we come up with about 180 peptides spanning more than 30 genes that have the right properties for therapeutic target. We see a lot of exciting new targets in this set, but also we find additional targets from well-established oncofetal antigens. So here you see a protein called FOX2B. It's a classical oncofetal antigen in neuroblastoma. We can reveal additional peptides restricted by B1501 and AO301. Here you see PRAME, a classical oncofetal antigen expressed in ovarian cancer, neuroblastoma, and many other tumors. We reveal additional epitopes from CO303, and SOX11 is another target that we emphasize. And around the many cases of cold tumors that we've studied so far, we can consistently reveal relevant therapeutic targets from well-established oncofetal antigens, in addition to novel targets that we're in the process of developing targeted approaches. Okay, so for this part, I'd like to conclude and summarize that using engineered chaperones, we can restore HLA production across many different cell states where low HLA expression is a hallmark of immune evasion. This has applications not only in cancer, but also in many infectious diseases that are known to downregulate class 1 as an evasion mechanism. We've shown that tapasin trafficking beyond the ER, when armored against degradation by ERAD, can facilitate presentation of natively processed antigen, expanding the observable immunopeptidome. And what I haven't had a chance to talk about here, but is currently in preparation, is that through deep mutational scanning of chaperone MHC1 surfaces, we can identify high-fidelity variants with enhanced chaperoning and peptide loading function towards further enhancing the outcomes of our immunopeptidomics campaigns. So all these systems really help overcome what I like to call the elephant in the room, which is the low production, the intrinsically low production of HLA complexes in cold tumors. But once we have that, now we have a roadmap and a set of targets for developing therapeutics. So there's one more challenge that we need to overcome, and again, there's very little need to belabor the point to this audience here, but we know that HLA molecules are incredibly polymorphic. Different HLA allotypes have polymorphisms in the peptide binding group, which means they present different peptide repertoires, which means that the name of the game is to identify peptides that can be presented in a conserved manner across many different common HLA allotypes. We call this public antigens. But even when we have those public antigens in hand, the problem is not solved because existing T cell receptors and TCR mimicking antibodies alike can only recognize peptide targets in the context of a very narrow set of HLAs. So this is the known term of TCR HLA restriction. And this is great for immunology, but from the perspective of drug development, this also means that separate therapeutic TCRs need to be tailored to the HLA genotype of each patient, which makes translation very, very difficult. So ultimately, what we want to do is, as soon as a patient with a particular indication walks into the clinic, sequence the tumor, identify public antigens, and the very first question we seek to answer is, can we determine the HLA allelic coverage or restriction of existing autologous TCRs or peptide-centric TCR mimicking antibodies targeting peptide HLA tumor antigens? This is an important question for being able to expand existing therapeutics. But also, in the case where we don't have available binders for particular HLAs, even though we know that the peptide can be presented, can we streamline the development of binders to address effectively any HLA present in the human population? And this is an area where really computational protein design and protein engineering can contribute significantly, as we found out in our studies. So I'd like to highlight a story through our own personal experience with neuroblastoma antigens on how we can effectively use these tools to really drastically expand the kids that we can treat with CAR T therapy. So this entire story began in the group of John Maris, who's very fortunately my next door neighbor at the Children's Hospital of Philadelphia. And John and Mark Yermakovits in his group identified a classical oncofetal antigen from a protein called FOX2B. You see here FOX2B expression is restricted to development. However, FOX2B is very robustly expressed in neuroblastoma tumors and is essential for the maintenance of those tumors. A peptide from FOX2B was identified to be presented across A2402, A2301, and multiple other common allotypes, making this a classical public tumor antigen for therapeutic targeting. John and his team went on to develop a TCR mimicking antibody called 10LH. And you see here 10LH recognizes FOX2B in the context of A2402, which was the parental HLA allotype that was found to be associated with this peptide. And deployment of this TCR mimicking antibody as a second generation CAR can clear tumors in xenograft models and is currently undergoing a phase 1 clinical trial at our children's hospital. So we wanted to ask the question, okay, we have seen this CAR, we have identified this CAR that can reach FOX2B in the context of A2402. What are some other possible HLA complexes that would be amenable to CAR T therapy using this CAR? This is a very relevant question as we're enrolling patients in this phase 1 trial. So we took a structural biology approach to answering this question. Here you see the co-crystal structure of FOX2B with A2402 in the apo form and in the complex with the TCR mimicking antibody. We can appreciate that the antibody docks very similarly to how a TCR would recognize the antigen. There's this very nice triad here from the CDRs that recognizes a key arginine from the peptide. But also when we zoom around the complex, we can appreciate there's a very important interactions between the CDRs of the antibody with what we call framework residues lining up the alpha-1 and alpha-2 HLA helices. These are highly polymorphic sites, and we reason that the exact identity of those side chains in HLA from different patients will dictate on whether 10LH could cross-react or alloreact, if you want, with different allotypes presenting the FOX2B peptide. So this is very helpful from one structure. We really got all the information we needed to go further and understand the HLA restriction of this TCR. So here you see the effect of alanine substitutions on specific polymorphic hotspots on the A2402 surface. And here you see the degree polymorphism of those residues across about 180 different HLAs, which are predicted to present the FOX2B peptide. Particularly in this element of the alpha-1 helix, we see here amino acids that follow the 3.6 residues per turn periodicity of the helix. These sites are highly polymorphic among HLAs that can present FOX2B. So instead of doing 180 experiments with all the different HLAs, we started introducing these polymorphisms piecewise on the parental A2402 structure to appreciate that some of these polymorphisms can be tolerated and still give binding to the TCR mimicking antibody. We measured this by SPR experiments, whereas other polymorphisms are pretty catastrophic for the interaction with 10LH. We see no binding by SPR for very specific reasons if we go back and model these substitutions based on the crystal structure. So we used all these single amino acid mutation data to build a very simple empirical model for ranking all the alleles that are predicted to present the FOX2B peptide, on one hand, according to a penalty that considers the effect on individual amino acid substitutions, and on the other, just based on the raw sequence similarity to the parental A2402 interface. And from this analysis, we can already separate a cluster of HLAs, A2402 and its micropolymorphisms, A2301, A3801, all of which are suggested to cross-react or alloreact with the 10LH TCR mimicking antibody. And when we go and test individual points from here, we find that they interact, albeit with a range of affinities. And when we test different HLAs from this group, selectively, we find that there's no interactions due to the presence of very specific polymorphisms right at the interface with the TCR mimicking antibody. And when we go also and test this in vitro using cytotoxicity assays on cell lines that are positive for, let's say, A2403, predicted based on our structural and biochemical assays to cross-react with 10LH, we see very robust killing using our second generation 10LH CAR-T. So this is all great because now we have a roadmap for understanding the alloreactivity range of the 10LH scFv and enrolling patients into the phase 1 trial. And the very nice thing about being at a children's hospital is that we have genotyped patient data from spanning several years, and we can estimate that the total combined patient coverage that would be amenable to CAR-T therapy using this TCR mimicking antibody is about 24%. So this is very encouraging, but at the same time, we can appreciate that there's other common alleles, and CO702 here is a prime example, that would be amenable to CAR-T therapy because they can bind and present the peptide, however, are not recognized by this existing CAR. So we wanted to build binders, streamline the development of binders for all the alleles presenting the FOX2B. And to do this, we started first by characterizing the basis of HLA restriction. So here we developed a method for analyzing polymorphic sites on the HLA surface. So this is informed by a database of TCR peptide HLA structures. We look at surface residues and appreciate that we have polymorphic hubs and conserved residues as well. And we can use this information to classify or cluster HLAs presenting similar surfaces in what we call T cell cross-reactivity groups in analogy to the established serological cross-reactivity groups that are commonly used in transplantation. And we find here that using this TCREG classification, we can in fact bind together A2301, A2402, and all of the other micropolymorphisms that we identified using our biochemical studies to cross-react with 10LA. So here we have provided a system where without having to do all this work and solve structures and do all this biochemistry, we can right out the gate have reasonable guesses for which HLAs would be amenable for expansion of therapy using existing TCRs and TCR mimicking antibodies. So back to those targets, those HLAs that we cannot target using the existing TCR mimicking antibodies, we also developed an experimental system. So here you see a bead-based assay. The beads are color coding. They're containing 97 different allotypes. And when we come in with a tetramerized form of our TCR or TCR mimicking antibody, we can exactly identify those HLAs that would cross-react with that particular therapeutic. So for the 10-alates scFv, we get back the same answer, A2402, A2301, A2403. When we apply this to different recombinant TCR systems, we also see this very strong restriction of interactions where any given TCR or TCR mimicking antibody will recognize a couple of common HLAs by not others. And this is really a unique feature of TCRs, where the TCR repertoire as a whole can, in principle, recognize any HLA. However, individual TCRs are restricted to a very narrow set of HLA allotypes. And how TCRs accomplish this is by using a very variable docking mode to anchor themselves on the MHC1 scaffold. So through pivoting and turning and adopting different docking angles, the CDRs of different TCRs can recognize different polymorphic sites and accommodate all the HLAs present in the human population. So we wanted to move away from that and develop a system that employs a rigid docking mode to recognize any possible peptide HLA combination. And this was from the perspective of being able to streamline the development of binders that would reach peptides irrespective of the HLA that presents them. So when we build our wish list for what the ideal peptide MHC antigen recognition platform would look like, we want it to be compatible with all the HLAs and peptides that can present. As opposed to T cell receptors, we want to have a rigid peptide-focused antigen recognition mechanism, and we want to be able to make this programmable. By introducing a limited number of amino acid changes, we want to be able to repurpose a scaffold for reaching different antigens. Of course, we want them to have high specificity and binding affinity, low immunogenicity for their developability into different modalities. And this is what we came up with in collaboration with Po-Soo Hwang at Stanford. We developed the first programmable protein engineering platform that allows us to really do exactly that. Here you see this tracer binder, which is inspired by superantigen, uses a helical frame that docks right on top of the peptide, and recognition of the entire length of the peptide is mediated via key side chains, specific 8 side chains at the tracer interface with the peptide antigen. This scaffold is non-immunogenic in animals, as we've found using both serological responses and ELISpots, and seem to have all the right features for developing it into robust modalities. So when we zoom in at our crystal structure of the first complex between an NY-ESO antigen and a proof of concept tracer, we see that the unique feature of this scaffold is where the tracer helices dock onto the HLA helices. These are some of the few conserved sites across the possible universe of polymorphic HLAs in different allotypes. So by leveraging a conserved docking mode that focuses on these sites, we reason that we have a scaffold that is compatible with all the HLAs present in different patients. So we went ahead and tested the specificity of our tracers. Here we used a technique that we developed in the lab for doing peptide exchange and developing libraries of tetramers. So here we tetramerized MHC, this is A0201, with 100 different peptides containing individual single amino acid substitutions of NY-ESO. Here we focused on the outward phasing residues of NY-ESO. And we compare the specificity pattern that we get for the tracer, shown here in the bottom, where with dark colors we designate cross-reactivity. We compare this cross-reactivity pattern to that of an engineered TCR called 1G4 that is very well known and established in the community to recognize the same peptide in the context of A0201. and appreciate that the tracer has just as good, if not better, specificity than the T cell receptor. We can also deploy this tracer as a second-generation CAR, and here we see potent on-target killing of lymphoma cell lines expressing the target NY-ESO, but not lymphoma cell lines that are not expressing NY-ESO. So it seemed like we were all in the right part, but really the great benefit of this platform is that it's programmable. So specifically, we reason that we can address the entire set of peptide and HLA allelic combination simply by reprogramming this 8 amino acid patch that interacts with the peptide in our crystal structure of the first tracer complex. And to show this, we took a selection of targets. So here you see different peptides spanning different HLA alleles. And through a very streamlined process by yeast display, we can develop and enrich a population of yeast clones that recognize either of these targets, but don't cross-react between them. And when we look at the features of these 8 residue diversified peptide recognition box on the tracer sequence and model the HLA complexes, we see good biochemical complementarity with the side chains of each peptide. So to summarize, we really have a specific programmable recognition across the entire peptide length. We have compatibility with different alleles. And through this reprogramming of the antigen recognition interface, we can develop really a family of biosimilars where we can reach many different HLAs using a common scaffold. So we wanted to test this, and we went back to this FOX2B example that I presented earlier to see if we can use this platform to address really that orthogonal set of the population that is negative for HLAs that would be recognized by the 10LH CAR. So we can quickly see when we analyze here different alleles that are presenting the FOX2B peptide that the majority of allelic coverage is made up of C alleles. And when we use our TCREG classification to look at the different C allele opportunities that can be recognized by different binders, we see that we have here CO702, CO701, and CO401 presenting very different surfaces, molecular surfaces relative to A2402. That could help us reach an additional 32% of US patients. So this seemed to be the right target for developing tracers. So first, we developed a tracer for FOX2B presented by A2402. This is the same exact allele that's recognized by the 10LH CAR. We use this as sort of a stepping stone to develop the tracer platform, having a positive control, which was the 10LH CAR. And we see here we get very little bystander killing using these second-generation tracer CARs. We get robust on-target killing of neuroblastoma cell lines that are either being transduced to express the target or are endogenously expressing the target with an efficacy that's comparable to the 10LH CAR. But the real benefit of doing this with a tracer is that once we have one binder, we can rapidly reprogram it to see a different HLA. And this is what we did here through the process I described previously. Diversification of this peptide antigen recognition box, we can develop a new configuration of side chains that now see the same FOX2B peptide in the context of CO702, a completely different HLA allele, while maintaining high specificity against the target, as we see in these experiments where we pulse monoallelic cells with the peptide and we follow binding with tetramerized tracer. And ultimately, we tested this novel CO702 binder for killing as CARs. And we see here no bystander killing, good on-target killing for cell lines that are expressing the CO701 or now more recently CO702 allotypes. And we also see that we can drive this killing even further through the expression of a single chain HLA construct to enhance antigen density levels, suggesting that killing is on target and everything happens for the right reasons. So for the second part, we have developed a new set of tools for therapeutic expansion. We've shown that the HLA restriction patterns of existing TCRs or TCR mimicking antibodies can be derived from an analysis of HLA framework residues for allogeneic therapy expansion, and we have developed a programmable protein engineering platform for developing binders for CAR T therapy. So with this, I would like to end. Acknowledge my lab and all the people that work on this project, our collaborators, funding agencies. Thank you very much for the opportunity to present, and happy to take any questions.
Thank you so much, Nick. It was really wonderful. I really enjoyed your presentation. We have just a couple of minutes for questions. There were actually a couple of questions about whether the presentation will be available later. Possibly yes, on my YouTube channel, but first I need to check with the speaker after editing, and eventually in 1 week time or 10 days time, hopefully it will appear in my YouTube channel. So any questions from the audience? Yeah, perhaps I can start. I was really impressed by these observations that you made, in particular in the second part. The figure that you showed, on y-axis you had this penalty scores, and then in x-axis cross-reactivity. So my question is that those HLAs with allo cross-reactivity, what are the correlates of allo reactivity? Perhaps you said, but I missed that. Is it structural with the configuration? Is it sequence features, or what is it?
So it's actually both. It's basically having the same amino acids at the key hotspots that are recognized by the TCR. So it will, of course, depend on the exact TCR docking mode. But when we take sort of a mean field approach, where we analyze all the structures together, we can come with this very strong sequence patterns that can be used to functionally classify HLA into Tregs. So without having to do any experiments, we can already get a reasonable list of sets of alloreactive HLAs for a particular TCR.
Yeah, just one. Another question is from Martin Flameye. Does cell surface tapasin interfere with TCR core receptor binding?
Yeah, that's an excellent question. And we had the same concern, Martin. It turns out it doesn't. If anything, it helps. We see enhanced killing and antigen recognition by T cells and peptide-centric CARs alike when we express tapasin TM. And structurally, tapasin would, of course, interfere with TCR binding because it does has a long N-terminal lobe that sits right where the TCR would bind. However, what we think is happening is that these clusters here that we're visualizing are dynamic. Tapasin molecules are diffusing in and out and stabilizing the MHC, and at any given moment, you still have enough binding sites, free MHC binding sites, for productive T cell signaling to occur. And it also helps that the dimensions of tapasin fit well within the immunological synapse, may contribute to additional phosphatase inclusion, and other things that are very important for signaling, as we know.
Thank you so much. I think we should stop here because the room that Nick and I are using for this webinar is booked. It was a fantastic seminar, and please join me to thank Nick again, and see you in my next episode. Thank you, everyone.